At its most fundamental level, the goal of a microscope is to magnify an object. The easiest way to do that actually is not with a microscope, but with just a magnifying glass, which is one single lens that can bend light to magnify an object. And if you've ever used a magnifying glass, then you probably know that they're really finicky tools. You have to hold them at just the right spots. You have to put your eye in just the right spot. And depending where you hold it, things get bigger and smaller. They go out of focus. And these are small annoyances for everyday kind of exploration. But if you're trying to do rigorous, reproducible scientific experiments, these are extremely problematic. Thankfully, in the 1500s, it was discovered that if you take two magnifying glasses and you stick them at a fixed distance from each other, you can get rid of all of these problems. And this gave way to what's known as the first compound light microscope, which has now become an indispensable tool for biologists, where they can visualize samples developing over time at up to 100 times magnification. An interesting thing about light microscopy is that you're never actually looking at your sample itself. What you're seeing in the microscope is how light interacts with your biological specimen. And that can happen in a variety of ways. Light can go directly through it, it can slow down as it passes through a sample, it can scatter off of it, and it can even transfer energy into your sample. And each of these different interactions can be used to develop a new type of light microscopy, things like bright field microscopy, fluorescence, or even confocal, which is a type of fluorescence microscopy. So now biologists have a whole suite of tools available to them to explore their sample with light microscopes. The light microscopes that we're most familiar with all use eyepieces to observe our sample. And in the early days, fantastic scientists like Santiago Ramon y Cajal would look through the eyepieces for over hours and intricately draw everything that they saw in the microscope. Now, modern microscopes have supplemented those eyepieces with digital cameras so that you can record images in 3 dimensions or even over time of your sample as it's growing, moving, dividing, or doing all sorts of things. This lets scientists not only have digital representations of their samples, but it allows them to make measurements about what they're doing. So you can say with very high accuracy how fast a cell is moving as it goes from one place to another, or how the shape of certain organelles change as a cell divides, for example. And this has catapulted biological research from purely using microscopy as an observational tool to being able to do rigorous quantitative, data-driven science with a microscope.